The cerebellum is the second largest region of the human brain. It is in the posterior of the head, in the occipital region, beneath the occipital bone. As you can see here, it covers the fourth ventricle, so it is easy to locate in that regard, and it lies over the brainstem with the medulla, the pons, and the midbrain. As you can see, the surface, known as the cerebellar cortex, has a large number of these folds known as folia. Um, so that this greatly increases the surface area. And if you notice, the gray matter was in the first couple millimeters of that cortex. So as we'll see stained in some of the upcoming sections, notice the difference in color, that the white matter is located in the interior of the cerebellum, but that the outer couple millimeters, the cerebellar cortex, has this greatly increased surface area. So that's far more neurons, which are possible given the folds. And so here, the white matter of axons has been stained, so it stains a darker color. And here, the lighter pink, you can see the gray matter. And notice that while there is a central region of the cerebellum known as the vermis, in Latin that translates as worm, the majority of the gray matter is located in these lateral cerebellar hemispheres. And notice that the folding of these cerebellar hemispheres greatly increases the surface area. We could split the surface of the cerebellum into a number of lobes. So there is an anterior lobe, which is separated from the rest of the cerebellum using this primary fissure. The largest of the lobes is known as the posterior lobes, and there's a couple of fissures, including the horizontal fissure. And then the smallest lobe, the flocculonodular lobe, is not visible from the surface, but you saw it from the midsection. And also here you can see the flocculonodular lobe of the cerebellum here. So there is a central vermis region, and then the lateral cerebellar hemispheres then have those separate lobes. As I'll show in another video, the cerebellum is attached to other regions of the brain through bands of white matter known as the cerebellar peduncles. And when one looks at these, one can see that these are axons, which obviously then you can stain in different colors there. So the interior of the cerebellum is mostly white matter, these axons here staining blue. And the gray matter with the soma are mostly located on the cerebellar cortex, which is highly folded. In addition, however, there are some deep cerebellar nuclei which also have gray matter. So while the majority of the gray matter is in the outer cerebellar cortex, there is deep gray matter soma located in the cerebellar nuclei. The largest and most obvious is this highly folded dentate nucleus. But note that there are then also others as well. So there is an emboliform, a globose, and a fastigial nucleus of the cerebellum. So once again, most of the gray matter is outside in these folds, but some of it is deep as well. What does the cerebellum do? Well, given that it's the second largest part of the brain, it does a lot, and perhaps we have underestimated its role. One of the difficulties is that, when we're measuring brain activity, the cerebellum, typically has some level of activity. So in addition to, say, movement, there's activity when we are thinking, learning, et cetera. And so the degree to which the cerebellum may play roles in a variety of activities, is being investigated. But one of its major role is that of motor coordination. So while the cerebrum does have a motor cortex and a premotor area, which may give commands to skeletal muscle, these smooth movements have been accomplished thanks to the input of the cerebellum. One of the things which is required for smooth movements like those is for individuals to have an idea where their body parts are. And so even when you close your eyes, you can walk, you can stand, because your brain knows where your legs are, where your arms are, etc. And that is because there are proprioceptors. Proprio means self. So if you speak Spanish, proprio means self. So you have receptors which aren't detecting vision or hearing or pain or temperature, but rather they are detecting yourself, where your own body parts are, these proprioceptors. And they go to the cerebellum. In addition, the inner ear is communicating through the vestibular cochlear nerve, the vestibular portion, about balance. And the cerebrum is communicating what muscles are planned. So the cerebellum is in a unique position where it knows what muscle movements are planned, but it also knows where the body parts are and whether we are on balance or not. If you were off balance, the muscles you would use to take a step would be different from, if you were maintaining balance. And so it is the maps of the body and the idea of self, these proprioceptors, which help the cerebellum, measure position and balance, and thus allow for smooth muscle movements. As I said, there are maps of the body. So if we were to imagine the cerebellum of a man or a woman, that there would be a map of the body looking something like this individual here, which would lie along the midline in the vermis region. Notice that the trunk and the head would be along the midline and the limbs would be lateral. And then there would be two representations of the body in the posterior lobe. So I'm sorry, the first in the anterior lobe, the second in the posterior lobe. So we have these maps of our body. So the cerebellum knows where the body parts are, and thus can help the cerebrum plan movements. The cerebellum can prevent overshoot. So if the cerebrum, let's say you want to put your brakes on in the car, your cerebrum might plan to use too great a force with too many motor units in the muscle, whereas the cerebellum would then limit those to only the appropriate number of motor units. So before one learns how to walk or drive a car, when an infant takes a step, or when you put on the brakes for the first time, you often use too much force. Often by memorizing what muscle movements are next, while a certain motion is engaged, the next one could already be planned, once again allowing for smooth muscle movements. The cerebellum is unique in its histological features as well. Once again, you can see that not only is the cortex highly folded, but so too are the dentary nucleus within the cerebellum. If you were to look at the cerebellar cortex, you would see that they were layers of cells here. So there is white matter that makes up most of the interior of the cerebellum. And then there is a granule cell layer, a Purkinje cell layer, and the molecular layer. Notice how densely packed the granule granule cell layer is. This is the densest accumulation of neuronal soma in the brain. In fact, not only do these represent the majority of neurons in the cerebellum, there are so many soma here that their numbers approach half the total estimated number of neurons in the brain. So this number of neurons is simply incredible. Nowhere in the brain are neurons as tightly packed as here. And then there is a monolayer of huge neurons called Purkinje cells. And there, so here you can see the soma of the Purkinje cells. Notice how much larger than they are compared to the neurons of the granule cell layer. And then there are a few cell types which can be found in the molecular layer above that. So I'm just going to quickly mention a few points, but I go into this a little bit more in the playlist. Once again, the granule cells, as we will see, they will contact Purkinje cells, and a large number of them, because they will send up an axon which then splits in the molecular layer to form a T shape, so that one of these can contact an enormous number. And as you can see, there can be well over 1,500 to 2,300 per cubic micron in the human brain. That's an incredible number of neurons. Nowhere else are as many nerve cells packaged in such a tight space as one sees here in the granule cell layer. The Purkinje cells, once again, form this monolayer of these huge nuclei. And these are the major way that the cerebellar cortex, the outside, communicates with the deep cerebellar nuclei. So these are the major output from the cerebellum. As we'll see, they inhibit the deep cerebellar nuclei with the inhibitory neurotransmitter GABA. So here you can see, these large Purkinje cells, and once again, in a monolayer, this layer is only one cell thick. There are other cell types as well. So there are Golgi cells, which are can be confined to the granule cell layer, while others project to the molecular cell layer. There are basket cells whose axons surround the soma of the Purkinje cells, forming a basket, hence the name. So if these were the Purkinje cells, the basket cells would be surrounding them. And then finally, in the molecular layer, there are the stellate cells as well. So there are not that many cells in that outer molecular layer of the cerebellum. The Purkinje cells, they are huge, and they have a dendrite tree which is flattened. And so here you can see the Purkinje cells, and I will try to animate this in 2 dimensions here. Right. So while they have all of these dendrites, they are flattened and all run in parallel. And then the granule cells, they then form those T-shaped axons known as parallel fibers, so that now each one can contact many of these Purkinje cells as it runs along the cerebellar cortex. So there you can see the parallel fibers there. Right. And so these Purkinje fibers, they have information coming in. So you can have climbing fibers coming from the medulla, like the olivary nucleus in the medulla here. So information reaches the Purkinje cells, and then also the granule cells. Both of these can stimulate the Purkinje cells, while then there are other cell types, such as the basket cells, which are making the inhibitory neurotransmitter GABA, the stellate cells, which are secreting taurine, and the Golgi cells, which can then interact as well. So with these incoming excitatory signals and inhibitory signals, then the Purkinje cells decide to go to action potential or not. And then the axons of these Purkinje cells will then project to the deep cerebellar nuclei, where they can inhibit neurons here and thus limit the muscular actions which would result. Okay. And so from the cerebellar cortex, the Purkinje cells would then inhibit those soma in the deep cerebellar nuclei. And so one of the things that's then striking under the microscope is how unique the cerebellum is, and with these various cell types, how these cell types are interacting with each other. And I have a bit more going through those cell types. Finally, the cerebellum is obviously connected to other regions of the brain, and where the white matter tracks reach the cerebellum, there are what are known as cerebellar peduncles, which then connect the cerebellum to other regions of the brain. So here you can see the cerebellum here, but here, whether it be the medulla, the pons, the midbrain, the cerebellum is then connected to those. And so there are superior cerebellar peduncles, middle cerebellar peduncles. One can observe them from the lateral side, but they also form the lateral border of the fourth ventricle. And in these peduncles, depending on where they're coming, there are different types of fibers which are leaving the cerebellum as efferent fibers, or afferent fibers coming into the cerebellum. And so there are these different cerebellar peduncles which then allow communication coming from the midbrain, the pons, and then with the inferior peduncles, cerebellar peduncles, the medulla. One final note is that the cerebellum has certainly changed over time and has been important, just like the cerebrum and other brain regions, in the evolution of jawed vertebrates, of tetrapods, of amniotes, etc. Now, we often give attention to the expansion of the cerebrum, going from, say, the condition of regular mammals to primates, or primates to apes, regular primates to apes, or apes to humans. But the cerebellum increases by about the same degree. So not only is the cerebrum of humans about 3 times larger than that of, say, a chimpanzee, the same would be true of the cerebellum. And so there are the cerebellum's increase in mass and changes there are probably important in many of the things which have made us human, such as upright walking or speech. And so this is probably very important in our ancestry, even though we often give more attention to the cerebrum.